nurses, and I'm happy to participate in this venture. So I'll be describing a little bit of background in genetics, and then some of the genetic principles that will help you understand the next lectures that will be given during the course. So beginning with history, genetics began as an observational study. This is Gregor Mendel working with studies on peas and other plants. His work has enabled understanding of general genetic principles, but it was only an observation. And when referred to humans, we could see a phenomenon here. The phenomenon is possible outcome of poor taste in dressing. So you could see that the parents have stripes. They were either vertical or horizontal, and there are different outcomes for possible children. Both stripes appear, none of the stripes appear, and so on. So these are observations. And in the past, genetic was only able to predict with statistical probability what are the chances of various outcomes of genetic conditions and different traits. However, with the discovery of DNA by Watson and Crick, and with the understanding that DNA is present in all cells of our body, and the DNA actually encodes our genetic makeup and also our traits and risk of having certain conditions or disease. Now, genetics became an interventional discipline, and I think that IVF and pre-implantation genetic testing is the ultimate intervention using genetics for the betterment of human health. So how is the genetic component of humans and other creatures, how is it arranged? So it'll be easy to look at this kind of a paradigm. We think of the human genome present in each cell as an encyclopedia. And of course, those of you who are old enough to remember that encyclopedias used to be in actual books and not Wikipedia on the web. But just imagine that each cell of the body has 23 chromosomes, 23 pairs of chromosomes. Each chromosome is just like a volume. And just like each volume in an encyclopedia has different chapters, so does the gene, so does the chromosome have different chapters. So a chapter in the chromosome is called a gene. It's actually a recipe for making a product that the body needs for proper development. Usually it's a protein. And in each cell of the body, we have about 20,000 different genes. And they are distributed along the chromosomes. And each chromosome has several hundreds to several thousands of different genes. The genes are made of DNA pairs, DNA base pairs. They are actually chemical letters. They could be either A, C, T, or G. And this is just a short segment of DNA showing the different letters that make up the genetic code. Within our genome, there are 3 billion letters, 3 billion base pairs, but most of them don't encode any product or protein. And these are DNA, it used to be called junk DNA because we didn't know what they did. Let's not call them that. We just say part of the genome, we don't know what the role structure is. But about 1 to 2% of the genome actually encodes the genes that make up the different proteins of the body. This part of the genome that encodes the proteins is called the exome, and it constitutes only about 1 to 2% of the entire genome. So now we have 23 pairs of chromosomes with a total of 20,000 genes written with about 3 billion base pairs. Okay, the genes are then transcribed into messenger RNA. The genes are in the nucleus of the cell, and the body makes copies of the different genes for production. So this is the recipe here in the DNA, and then using an enzyme called RNA polymerase, a short fragment, a short copy of that gene is made. This is called the messenger RNA. By the way, Moderna and Pfizer vaccines for COVID are using messenger RNA, but not human messenger RNA, but the messenger RNA of the coronavirus. The messenger RNA is then moved into the part of the cell called the cytoplasm, where the ribosomes make the proteins. So the messenger RNA is actually a template. It's actually a code for making the different proteins, and the messenger RNA is read by a small molecule called the ribosome, which actually creates the protein, which is a long strand of amino acids. And these proteins are actually the makeup of different parts of the body. So they could be structural, like collagen or elastin. They could be hormones, or they could be receptors, and so on. So alterations in the genetic code can cause different aberrations, different genetic conditions, different diseases. And they can be divided into several layers of hierarchy, depending on where and how large the aberration is. So beginning with the most basic and most robust changes, these are the chromosomal changes, either an extra chromosome or a missing chromosome. Then there could be smaller changes, like deletion of a part of a chromosome. And finally, they could be sequence changes like mutations. And we'll go over each one of them and give examples. So chromosomal problems have been known for decades. And the way to detect them used to be quite simple. Using a microscope, we could actually see the chromosomes in each cell. The technology for looking at the chromosome has not changed over the last decades. And we can see an extra chromosome or a missing chromosome. For instance, Down syndrome, also called trisomy 21, is caused by an extra chromosome 21. So usually there should be 23 pairs of chromosome. In Down syndrome, there are 23 pairs and an extra chromosome 21. So overall, not 46, but 47 chromosomes with an extra chromosome 21. And there are other examples of chromosomal problems like trisomy 18, that's an extra chromosome 18, and so on. Also, there could be sex chromosome abnormalities like an extra chromosome X in a male or an extra chromosome Y, and so on. So different chromosomal problems. These could be found using a microscope. However, sometimes we find an abnormality that is present only in part of the cells in the body. Usually, either all cells are normal or all cells are aberrant with an extra chromosome, but sometimes we find a condition called mosaicism. In mosaicism, we have different kinds of cells in the body. Some of them could be normal and some of them could be abnormal, or they could have different abnormalities. For example, Turner syndrome is usually a missing X chromosome in a female, but Turner mosaic could have some of the cells with 45X, a missing chromosome, and some cells with a normal composition. So mosaicism is not extremely rare. It is particularly common in pre-implantation embryos. This is why it's important. And the consequences of such mosaicism depend on what chromosomes are involved. Some chromosomes are more harmful than others. For instance, chromosome 21 for trisomy 21 is more harmful because they could result in the birth of a child affected with mosaic trisomy 21. So this is a mosaic Down syndrome. Other chromosomes are not as harmful. Also, it is important to know what is the percentage of abnormal cells in the embryo or in the body. If the percentage of abnormal cells is low, the impact is probably going to be quite mild. However, if a greater proportion of the cells are abnormal, it is more likely to cause an effect. Then the distribution of the abnormal cells is also important, because sometimes the distribution is uniform throughout the placenta and the body of the developing embryo and fetus. And sometimes we find the abnormality only in the placenta, whereas the fetus is normal. And also there are other problems with this. So this is the first type of abnormality. They're called numerical because there is an extra or missing chromosome. Sometimes, however, there are structural abnormalities in the chromosome, for instance, translocations. Just imagine that the orange and purple chromosomes are normal chromosomes, but sometimes a translocation occurs wherein part of one chromosome is transferred onto the other and vice versa, creating a balanced translocation. Individual with a balanced translocation is usually normal because there's no added or missing genetic information. So when all the genetic information is present in the body, there are usually no consequences. However, when such an individual procreates with a normal individual, sometimes we get a condition of an unbalanced translocation, where, for instance, here, there is extra information from the chromosome marked in purple and missing information from the chromosome marked in orange. And this may result in infertility or in early miscarriage. And if miscarriage has not occurred, sometime in the birth of affected offspring suffering from malformations, mental retardation, or both. Next we find other genetic abnormalities that are submicroscopic. They're so small that the microscope cannot detect them. And they're usually thought or considered either deletion, a small part of the chromosome is missing, or duplication, a small part is added. And here are some examples of common genetic condition caused by microdeletions. This is one of the most common. It's called DeGeorge or velocardiofacial syndrome. This syndrome is caused by a missing part of chromosome 22. The part that is missing is only 3.5 million base pairs or million letters. This is too small for the microscope to detect. And there's some other conditions like Williams syndrome or Prader-Willi. These are all called microdeletion syndromes. And in the past, we thought there were quite a few, but now we recognize that there are many, many different microdeletion syndromes. And these can be detected using technology called microarray or comparative genomic hybridization. With comparative genomic hybridization, it's called also microarray or genetic chip analysis. Usually, we take the sample from the tested individual. It could be from a child, it could be from a parent, it could be from a fetus in utero, or it could also be taken from a blastocyst. We take some cells, we extract the DNA, and then we label the DNA from the patient with a fluorescent green dye. We take a reference DNA marked in red, and then we hybridize them to a microarray. It's an array that contains thousands or hundreds of thousands of different probes, each targeting a different part of the chromosome. And using this, we can detect whether there's an extra chromosome, a missing chromosome, or actually even a deletion or a duplication in a part of a chromosome. And this is important because this technology is now used in pre-implantation genetic embryos just to detect aneuploidy, meaning to detect chromosomally abnormal embryos. So this technique has now taken over in doing a lot of technologies called PGTA, pre-implantation genetic testing for aneuploidy. It's also important for couples where in one of the parents carries translocation, a balanced translocation. And so this is quite a useful technology. And with this technology, we can detect different abnormalities, and the computer tells us if there's 3 copies, which is a trisomy, 2 copies, which is normal, or a missing copy. Finally, diving even deeper into the genome, we find that some changes, some alterations are at the single nucleotide level. So amongst the 3 billion base pairs, some conditions are caused by an alteration in just one of these letters. And this is called a mutation or a sequence variant. In the past, we used to think of them quite simply as either being a mutation or a normal variant. But now we are forced to see these many different changes and look at them as a spectrum of different changes where some of the changes or the variants may be considered pathogenic, what we would call in the past a mutation. Some of them are more likely to cause, but not in 100%. Some of them are variants of unclear significance. We're not sure what they do. And some variants, most of the variants, are benign. They don't cause any problems. So not every change or not every variant in the genetic code is harmful. There are a lot of them which are benign, don't cause any problem. And I'll give you some example. For instance, if this is the genetic code, this is a small part, and this one letter, the A, changes to a T. This may be like a change in the spelling, but it's not a misspelling, just different ways of spelling the same word, like gray and gray. This is called can contain tens or hundreds of different genes, just like a little byte. And then we can look at a single gene. This is a small little portion. Now, in the past, we needed to check each gene individually and each mutation individually. Currently, we have technologies that are called next generation sequencing or massive parallel sequencing. And this enables us to analyze in one single test, to analyze the entire exome, which is 1 to 2% of the genome, and even the entire genome. So currently, if we have a suspicion of a genetic condition, we can use these robust technologies and come up and find different mutations in different genes and analyze different problems in the family. So for instance, if the parents already have an affected child, or they've had termination of pregnancy for an affected fetus, it's not a good idea to say, well, this is just a chance, let's try again. It's possible now in many cases, up to 50% of the times, to find out what the genetic cause was. And if this is something hereditary, if it's something that has a chance of recurring, we can use this information and avoid birth of affected offspring using pre-implantation genetic testing or prenatal diagnosis during an ongoing pregnancy. So many of the children with abnormalities or fetuses with aberrations or malformations are caused by different pathogenic variants in different genes, and these have different modes of inheritance. So inheritance depends on what gene is affected, what its role is in the body, and how it is transmitted through the generations. So a simplified way is looking at mom and dad, looking at the different facial features and saying who the child looks like. But of course, when we're dealing with 20,000 genes and when there's so many mutations, this becomes a little more difficult. So I'd like to give you some ideas of different modes of inheritance. The first type of inheritance is called autosomal dominant. So this is a typical characteristic pedigree of a family with an autosomal dominant condition. I'll give you some examples later. In this family, we see males in squares, females in circles, and then the shaded circles or squares are affected individuals. So you can see that this individual here in the first generation is affected. Line across means that the person is deceased. Now we see this person has several children. Some of them, half of them are affected, half of them are not. This is the spouse of this individual, and this individual also has both a son and an affected daughter. So we can see that the condition is transmitted to half the offspring, regardless of their gender. This is called autosomal dominant. Now, looking a little deeper into why this occurs, we know that each individual has 2 copies of each gene. And say, in this case here, looking at a family here, let's say the mother is unaffected, and this is the chromosome, and this is the gene within the chromosome. The mother has 2 copies, and both copies are normal. And the father, who is affected with an autosomal dominant condition, has 1 normal copy, but 1 abnormal copy is enough to cause the condition. Now let's see what the possible outcomes of offsprings are. If the mother will always pass on a normal copy of the gene, so this is always the maternal copy marked in kind of yellow. The father may either transfer the normal copy or the abnormal copy. So half of the children will have the abnormal copy from the father, and half the children will have the normal copy from the father. So regardless of sex, we'll have both healthy boys and healthy girls, and half of the children will be either affected boys or affected girls. This is called an autosomal dominant condition. It's called dominant because the abnormal copy is dominant. 1 abnormal copy is enough to cause the condition. And here are some examples. Neurofibromatosis is a common condition with different skin lesions. Achondroplasia is one of the common types of short stature, common type of dwarfism, and Marfan syndrome, and so on. There are many, many syndromes that are dominant. Another condition is called codominant, meaning that each type of variation is apparent. This is characteristic of blood types, blood type A and B. Since this is not a disease that will skip that. Many, many different conditions are called autosomal recessive. In an autosomal recessive pedigree, we have healthy families. Both parents are normal, but they may have abnormal daughters or sons. And then this doesn't happen again in next generation. The reason for such a condition is that both parents are carriers of an affected copy of the gene, of an abnormal copy of the gene, but they are healthy. They are healthy because it's a recessive condition, and having one abnormal copy is not enough to have the condition, because the normal copy compensates. Okay, so the father is a carrier having one abnormal and normal copy. Mother also is a carrier. Both are healthy, but the offspring may have different conditions. So if this girl has inherited both normal copies, one from mom and one from dad, she will be unaffected. These individuals have inherited one normal and one abnormal copy, so they will be carriers. They will be normal like the parents, but still being carriers. And in 25%, the offspring, whether it be a boy or a girl, has inherited one abnormal copy from the father, one from the mother, and will be affected. So with autosomal recessive conditions, 25% of offspring will be affected, 50% will be normal carriers, and 25% will be unaffected non-carriers. And there are many, many different examples of such conditions like cystic fibrosis, spinal muscular atrophy, Tay-Sachs disease, common in Ashkenazi Jews, thalassemias, common in individuals from the Middle East and Southeast Asia, sickle cell anemia, common in individuals of African descent, and so on. It's important to recognize that many of these autosomal recessive conditions are currently tested using expanded carrier screening programs. And I think that for IVF nurses who do the intake for many of the patients, it's important to understand that before undergoing IVF, and particularly before undergoing pre-implantation genetic testing, it's important that parents undergo at least basic, but preferably expanded carrier screening, which enables us to detect such carrier parents and avoid birth of affected offspring using pre-implantation genetic testing for monogenic disorder, for such disorders. In fact, a couple of weeks ago, the American College of Medical Genetics has issued a new guideline recommending that all prospective parents undergo expanded carrier testing. Expanded carrier testing, hundreds of different autosomal recessive conditions are tested at the same time, detecting carrier states in many, many different genes, and allowing to detect such couples where both partners carry a pathogenic variant, a mutation in the same gene, and are at such risk of having these serious genetic conditions in the offsprings. There are other genetic conditions that are passed on only by the mother. They're called X-linked recessive conditions. X-linked recessive conditions are associated with genes that are on the X chromosome. Males have only one X chromosome and one Y chromosome. So if this is a gene on the X chromosome, and if it's mutated, then this individual will be affected. So usually with X-linked conditions, the father is always healthy. But the mother could carry a mutation in such a gene, but still be normal because she has another normal copy of the gene on the other X chromosome. Remember, this is an X-linked recessive condition. And with recessive conditions, one normal copy is enough to keep you healthy. But such parents where the mother is a carrier of an abnormal copy of the gene, such parents are at risk of having affected sons. Why is that? Because the father will transfer the X chromosome to the girls, because when the mom always transfers an X chromosome, so X chromosome from the father and X chromosome from the mother will result in a girl in any case. If the mom transfers the normal X, the daughter will be healthy, unaffected, and not a carrier. And if the mom transfers the abnormal X chromosome, the daughter will be healthy but a carrier. If the father transfers the Y chromosome, it's a boy in any case. Now, depending on what the mother transfers, if she transfers the normal chromosome, then the son will be unaffected. However, if she transfers the abnormal X chromosome, this child here will be affected because he only has one X chromosome, but with the abnormal copy, and the Y chromosome does not compensate. So with such a pedigree, we will see only affected males, and they are linked to each other through their maternal lineage. So these two boys are the daughter of this woman here, who is a carrier, but she wouldn't know that. She would have a normal mother, but she may have affected sons or affected nephews by her sisters, but not through her healthy brothers. And here are some examples, like Duchenne muscular dystrophy is quite a serious condition causing muscular dystrophy and early death, the different types of hemophilia, and of course milder conditions like color blindness and G6PD deficiency. And in fact, Duchenne muscular dystrophy is now included in many expanded carrier tests that are performed. So I think with this, we can summarize and say that understanding genetics is important for all health care professionals, but particularly, I think, for individuals dealing with reproduction and IVF nurses. I hope you have enjoyed, and hopefully graduating from Genetics 101 will enable you to better understand the next lectures that will be coming through the course. Thank you very much.
Well, hi, I'm Dr. Silver from the Infertility Center of St. Louis, and we're going to talk on IVF Worldwide now about IVM, that's in vitro maturation of oocytes from ovary tissue.